Exploring Life. Chapter 8, Eukaryotes. Part 2. Now that we've covered molecule manufacturing and breakdown, let's move on to the second major type of vital process in eukaryotic cells, energy processing. This includes the work of two key organelles: mitochondria, which handle cellular respiration, and chloroplasts, which carry out photosynthesis in plant and algal cells. This image highlights key features shared by mitochondria and chloroplasts, organelles found in eukaryotic cells. Both contain their own circular chromosomes, have ribosomes distinct from those in the host cell, and are surrounded by double membranes. All these features support the endosymbiotic theory, suggesting that these organelles originated from free-living prokaryotes that were engulfed by an ancestral eukaryotic cell. Mitochondria are responsible for cellular respiration. Their main role is to convert nutrients into ATP, adenosine triphosphate, which serves as the cell's primary energy currency. Mitochondria have a distinct structural organization that supports their role in energy production. The inner membrane folds inward into structures called cristae. Cristae are the inner folds that increase surface area, giving the mitochondrion more space to carry out energy-producing reactions. They also house ATP synthetases, specialized enzymes that are essential for making ATP efficiently. This transmission electron microscope image of a mitochondrion shows key structures we've just discussed. You can clearly see the double membrane and the folded cristae of the inner membrane, features that are essential for efficient energy production. Chloroplasts are responsible for photosynthesis, a process that occurs in plant cells and some algae. Their main role is to convert sunlight into chemical energy, storing it in the form of glucose, a vital energy source for the cell and, ultimately, for the entire food chain. Chloroplasts have a specialized internal structure that supports their role in photosynthesis. The stroma is the fluid-filled space that serves as a central hub for essential metabolic processes. Within the stroma are grana, stacks of thylakoids that absorb light and convert it into usable energy during the light-dependent reactions of photosynthesis. Chloroplasts also have their own DNA and ribosomes, and their genes show strong similarities to cyanobacteria, further supporting their endosymbiotic origin. This transmission electron microscope image captures the internal structure of a chloroplast. You can see key features we've just discussed, including the stacked grana made up of thylakoids, as well as the surrounding stroma. We've already reviewed how cells manufacture and break down molecules, and how they process energy. Now let's look at the third and final major process we're going to talk about: structural support, movement, and communication. This is made possible by the cytoskeleton, along with structures like cell junctions and the extracellular matrix. These not only give the cell shape and stability, they also play a role in how cells interact with their environment and each other. The cytoskeleton has three main components, all embedded within the cytoplasm of the cell: microtubules, actin filaments, and intermediate filaments. This table helps visualize the differences between the three main components of the cytoskeleton. It compares their structure and function, showing how each type of microtubules, actin filaments, and intermediate filaments plays a distinct role in maintaining the cell's shape, movement, and internal organization. Microtubules are the thickest and strongest components of the cytoskeleton. Structurally, they're hollow tubes made of alpha-tubulin and beta-tubulin dimers, forming long, rigid filaments. Functionally, microtubules serve as tracks for intracellular transport, helping move vesicles and organelles around the cell. They also play a key role in chromosome separation during cell division, support cell shape and structure, and form the framework for modal extensions like cilia and flagella. You can think of microtubules like rigid rods. They give the cell strength and stability, and allow it to push and transport materials with precision. Cilia and flagella are modal structures made of microtubules, and they play key roles in movement and transport in eukaryotic cells. Cilia enable movement in unicellular eukaryotes like paramecium. In multicellular organisms, they help move substances, for example, by clearing mucus from the airways or guiding an egg through the fallopian tube. Flagella are typically longer and fewer in number. They provide locomotion for unicellular organisms like Euglena and enable sperm movement in animals. Unlike prokaryotic flagella, which are made of bare protein filaments and rotate like a propeller, eukaryotic flagella are extensions of the cell, enclosed by the plasma membrane and built from a precise arrangement of microtubules. This internal framework drives their characteristic whip-like motion. Actin filaments, also known as microfilaments, are the thinnest components of the cytoskeleton. They're made of the protein actin, which forms long, twisting chains. Actin filaments act like ropes. They're strong yet flexible, ideal for pulling, shaping, and enabling movement. They help maintain cell shape, are essential for muscle contraction, and play a key role in cell division, especially during the final step of cytokinesis in animal cells. Pseudopods, or false feet, are temporary extensions of the cell membrane, and their formation is supported by actin filaments. They play a vital role in cell movement, environmental sensing, and food capture, especially in organisms like amoebas. In multicellular organisms, like humans, pseudopods are essential for the immune system. Cells like macrophages and neutrophils use them to engulf bacteria and cellular debris, helping defend the body against infection. Muscle contraction relies on actin as well. It works together with myosin, a complex motor protein that binds to actin and pulls on it. Together, these two components slide past each other, shortening the muscle fiber. As a result, the muscle contracts, producing movement and generating force. Flex your muscle a bit, actin is putting in the effort. Intermediate filaments are made of diverse fibrous proteins, often tailored to the specific tissue type. Their main job is to provide structural support and mechanical stability, especially under stress. They help maintain the cell's shape, protect the nucleus by supporting the nuclear envelope, and anchor organelles in place. These filaments are especially important in tissues like skin and muscle, where physical stress is constant. One well-known intermediate filament is keratin, a major component of your skin and hair. So yes, you're literally carrying intermediate filaments through life and staring at them every day. This illustration shows how unicellular organisms, protists, use different cytoskeletal elements for movement. On the left, paramecium uses cilia, made of microtubules, to propel itself through water. In the center, amoeba moves using pseudopodia, driven primarily by microfilaments. On the right, Euglena swims using a flagellum, which, like cilia, is built from microtubules. Together, These examples highlight the diverse and adaptable movement strategies of unicellular eukaryotes. Both animal and plant cells are eukaryotic, but our generalized description of eukaryotic cells has been largely based on the animal cell. While plant and animal cells share many features, plant cells have several unique structures that set them apart. In plant cells, the cell wall provides structural support and protection. Chloroplasts are the sites of photosynthesis, converting sunlight into chemical energy. The central vacuole helps maintain turgor pressure and stores water and nutrients. Plasmodesmata are tiny channels that allow communication and transport between neighboring plant cells. These specialized features reflect the unique adaptations of plant life. Now let's look at features unique to animal cells. They have no cell wall, only a flexible plasma membrane, allowing for a wider range of shapes and movements. They lack chloroplasts, so they rely on consuming other organisms for energy. Lysosomes are present and contain digestive enzymes that break down waste and cellular debris. Centrosomes, which include centrioles, help organize microtubules and play a critical role in cell division. This illustration compares the distinctive features of a typical plant cell on the left and a typical animal cell on the right. The plant cell includes structures not found in animal cells, such as a cell wall, chloroplasts, and a large central vacuole. In contrast, the animal cell lacks these features but contains centrioles, which are not found in most plant cells. These differences reflect the unique functions and environments of plant and animal cells. Once again, it's time to pause and review. Let's check your understanding with a quick self-evaluation. Take a moment to reflect on what you've learned so far. Can you explain the key structures and functions of eukaryotic cells? Do the analogies and examples make sense? Use this opportunity to reinforce your knowledge before we move on.